Hello everyone, today we are going to see microbial carcinogenesis. That means microbes are involved in the causation of cancer. Now let us see oncogenic RNA viruses. RNA viruses example is HTLV1. HTLV1 is human T cell leukemia virus. It is the only retrovirus which is implicated in the causation of cancer. Okay, now which cancer? HTLV1 causes T cell leukemia or lymphoma. It has a tropism for CD4 positive T cells. And these CD4 positive T cells are the target for neoplastic transformation. Human infection. How does a human acquire this infection? For the infection, it requires the transmission of these infected T cells. Now, these T cells could be transmitted via a sexual route or blood products or by breastfeeding. Now, let us see the HTLV1 genome. Because it's a retrovirus, so it will contain all the genes which are typical of a retrovirus: GAG, ENV, and long terminal repeats. But it also contains another region also, that is TAX. And this product of this gene, TAX, is essential for viral replication. Now let us see this TAX protein. It's very important. One thing we have already seen, that the product of this gene is essential for viral replication. So the TAX protein, it will stimulate the host genes, and by the stimulation it is involved in the proliferation as well as the differentiation of T cells. So one thing, it is important, it is involved in the proliferation of the cells and also the differentiation of the T cells. Second thing is, it inhibits the cell cycle inhibitors. The cell cyclin inhibitors, that is P16INK4A. So it inhibits the cell cyclin inhibitors and simultaneously it stimulates the cyclin D activation. So it causes the dysregulation of the cell cycle. Okay. Other than this, it stimulates NF-kappa B. NF-kappa B is the transcription factor. It is the transcription factor for genes like pro-survival genes and anti-apoptotic genes. Okay. So by stimulating NF-kappa B, it is improving the survival and decreasing the apoptosis. And also it is leading to genomic instability, and how it is interfering with the DNA repair. Okay, so we have seen that it is implicated in the proliferation of the T cells. Also, it is inhibiting the cell cycle inhibitors like P16INK4A. And it is stimulating the cyclin D activation. Okay, so the cell will keep on replicating. And also it will stimulate NF-kappa B. So it will improve the survival of the cell, and it will prevent the apoptosis of the cell. And also it is leading to genomic instability. It is interfering with the DNA repair function. So this is all because of this TAX protein. Now let us see HTLV1 infection. What it causes? It causes the proliferation of non-malignant polyclonal cell population. So it causes proliferation. We have already seen it causes T cell proliferation. Now initially these are non-malignant polyclonal cells. Okay. And why are they proliferating? We have already seen this is because of stimulatory effect of TAX on the cell proliferation. Now these proliferating T cells, they have increased risk of mutation. Along with that, we have already seen that the TAX induces genomic instability. So this instability will allow the accumulation of mutation also, because the DNA repair function is also not occurring. So it will allow the accumulation of the mutation. So initially it causes the proliferation of non-malignant polyclonal cells. But now because of genomic instability, because of accumulation of mutations, now it will lead to emergence of a monoclonal neoplastic T cells. So this is how HTLV1 is implicated in the causation of T cell leukemia or lymphoma. It is the only retrovirus which is implicated in this carcinoma. Thank you.